DNA contains the instructions for all life on Earth. Everybody has a DNA sequence unique to them, and DNA is found in almost every cell in the human body. We call sections of DNA that code for specific proteins genes, but just because a cell contains a complete blueprint to create you doesn't mean it uses all of it. Let's think about it. You wouldn't want a cell, an eye cell specifically, to produce hemoglobin, which is needed for oxygen transport, or a heart cell to produce mucus like the cells in your airways do. You also don't want certain proteins being overproduced. Inside a cell is like a tiny factory producing things the cell and your body needs to function. And you can't have a factory producing too many things, right? That's where RNAi comes in. The I stands for interference. When a gene is going to be expressed, DNA gets transcribed into mRNA, which gets translated into proteins by ribosomes. Ribosomes are the chefs of your cell, making any kind of protein as told to by mRNA. The RNAi system comes in and stops mRNA from being translated by ribosomes. RNAi also stops the production of proteins from foreign RNA that may have come from a virus. When a cell detects long double-stranded RNA, a protein called dicer cuts it into smaller pieces. These small chunks of RNA bind to RISC, which stands for RNA-induced silencing complex, a complex being a group of proteins working together. One of these proteins, called ARGO2, rips the pieces apart. These pieces are compared with strands of mRNA. When a match is found, the slicer protein chops up the mRNA molecules, preventing the ribosome from producing proteins. No more mRNA means that there is nothing for the ribosomes to make. In the lab, RNAi is used by scientists to stop gene expression and see how it impacts organisms. There's also research being done to hopefully use it to treat things like cancer, viral infections, and many more.